Thank you, Malcolm, and thank you to Richard and the organizing committee for inviting me. It's great to be here and such a wonderful venue and with such a great audience. I was asked to talk today about sort of what we've been doing in terms of research networks and clinical registries. And for many of you, my name may be a new one, but I've actually been in the CFSME field for about 6 or 7 years now. I started out, my background is infectious diseases, and I started out at Stanford, and Jose and Montoya and I saw the first CFS patients together at Stanford and created the Valsite trial there, which was one of the first, certainly first in a long while, placebo-controlled, double-blinded studies in CFSME. But before that, I spent a lot of time looking at sort of what people would call host response or gene expression side of pathogenesis, and thinking about that. And even before that, I bothered to go get a PhD in bioengineering, or really computer science, and sort of vetted myself in bioinformatics and sort of study of large data sets, which turns out is turning out to be pretty useful for CFS. So I left Stanford about 2 years ago, and with a group of collaborators started the Open Medicine Institute, which, as Malcolm indicated, we're trying to put a lot of things together. But one of the key premises, and I think it's really underscored for CFSME, is that clinical medicine, particularly in the US, but I think tendency is worldwide, has divorced itself over time from basic clinical research. And the average in the US, the average physician sees a patient for about 10 minutes, which, as you all know, or many of you know, as ME patients, you can't get anything done in 10 minutes for anywhere near the kinds of symptoms that CFS patients have. But this applies to a lot of other diseases. So there's a lot of things sort of being swept under the rug steadily as we evolve through what is our clinical course these days. So our goal in starting the Open Medicine Institute was to bring that back. And it's not that physicians are not interested in doing research, it's just that they don't have the time and the resources anymore to do it. And we wanted to change that. We wanted to bring them an infrastructure for bioinformatics, for helping support their clinical workflow, and at the same time extracting out information so that they could use, that we could use, but they could also use in looking at research. Combining that with a biosampling infrastructure, so a biobank, and the ability to take samples from those same clinicians, from hospitals and so forth, to combining that with the latest in collaborations for providing the latest and greatest, as Malcolm indicated, in terms of biotechnology and the ability to process genomic and proteomic samples and so forth. Our institute is right in the heart of Silicon Valley, which these days is really more biotech valley, and that's served us really well, including collaborations still at Stanford and UC and so forth, and sort of up and down the valley, but outside the valley as well. And finally, we wanted to bring a social networking component to it, which is also something that's exploding in Silicon Valley, and I know many of you have used some of the outgrowths of that here and around the world in terms of Facebook and Twitter and things like that. But we wanted to bring some of those aspects to medicine, and I'll talk a little bit more about what we're doing there. So really, one of the problems with ME as a research subject, if you will, is this idea that it's a syndrome. And in medicine, we're really poor at dealing with those, both from a clinical perspective, but also from a research perspective. The secondary thing, as David very nicely gave an overview of, is that we haven't done a great job at sort of, and Annette as well, at defining those diseases. And part of the problem there is that the metrics are lacking. David just gave a wonderful overview of some of the metrics that he's used very skillfully over the years, but when it comes down to it, some of these instruments just are not very sensitive, and we need a better way of looking at those definitions. So how do we really take all that and use it and apply it to advance the practice of medicine and hopefully answers for our patients? So just looking at history, we've slowly been acquiring lots and lots more data around different diseases. And if you look at the last 10, 20 years, there's an explosion of the amount of data that some of these tests are generating, be it from gene sequencing, microarrays, imaging studies. I mean, fMRI gives us more, so functional MRI gives us more insight into more data points around of a patient's brain or other locations than an X-ray ever could have, in the past. And now we're looking, in 2011 and 2012, we're looking towards, whole genome sequencing being available and a host of other technologies that I could spend hours on. So, but the issue really still comes back to diagnostics. It's the typical blind men looking at the elephant. And, which part of the elephant, I'm probably the guy over there on your left. Which part of the elephant are you looking at? And we haven't really figured that one out yet for ME. We have all these people feeling around for different parts of the elephant, and nobody really has put their finger around it. And maybe there's two elephants in the room. Maybe there's an elephant and a monkey. Maybe there's a lot more than that. We don't know. So I think that's an important point to take home, not just in a joking way. But it comes back to, where are we on this pyramid? And this is sort of one of the classic pyramids that gets thrown at us in medical school in terms of what's the balance between genetics, between the environment. So are there other environmental stimuli that have caused problems for CFSME? People have brought up mercury, arsenic, other toxins, and then vectors, and how much exposure did you have to a particular agent, and is there a piece there? Well, we can look at all those in a lot of ways, and I think we've been looking at them actually in relatively limited ways so far in CFSME, in that we can collect data from observations, and that's largely what a lot of the work has been done in terms of both the assays that David just mentioned, but that's sort of the phenotype. Informatics has done a lot to help us collect those, and now sort of the biotechnology piece is adding more and more to the sort of the harder data in terms of samples collected and run under different technologies. So I'm not going to spend a lot of time on this because I think the previous speakers both sort of hit this, but just the point is really that there is a lot of overlap in CFSME symptoms. There's overlap into other areas. I gave a similar talk at the Lyme meeting last year, and we've made lots of inroads at developing more of a scientific network there as well. The disease definitions just aren't mature. At my first IACFS meeting, I was just stunned that 30 years into the disease, people are still arguing about a case definition. Well, that's a problem. We cannot move a field forward if we are not all talking about the same thing. And so we need to at least come to some agreement on some of those, even if it's not the right agreement. We need to just put a stick in the sand and move on, and allow some science to be built around those sticks. I'll talk more about the biomarkers in a moment. Treatments clearly are not standardized. People, I think David already alluded to this as well. Obviously, lots of people claiming that they have one treatment versus another that cures everybody. And my experience has been that the more people claim a treatment, the more of them show up at your doorstep, a different physician's doorstep, a year or 2 years later, and they just don't follow up with that physician who didn't treat them fully or well enough because they weren't satisfied with that, obviously, in the end. So there isn't that long-term follow-up, and that's really when it comes down to outcomes data. That's what we have to collect in this field is outcomes data, hard outcomes studied in a rigorous way, and that hasn't been the case for all too many studies. Back to the issue of sort of general medicine, I think we're sort of at a crossroads here, and CFSME is sort of caught in the middle of this conflict, and I think that's one of the reasons why we have so much difficulty getting a lot of attention to the field. Medicine has been heading towards this guidelines view, evidence-based, but guidelines view of medicine. So patient has X, a diagnosis X, therefore you do Y or Y, Z, and ABC. And this sort of cookie-cutter approach to medicine works very well for large, efficiency-oriented clinics that, the sort of the McMedicines of the world that push you in and push you out. But it's really a lousy system for things that don't fit inside the box. CFSME, unusual cases, things that are more complicated, and things that just simply take longer. And besides that, it stifles medicine. That it runs headlong into this revolution in medicine, which people are calling personalized medicine. Hopefully, for most physicians, medicine is relatively personalized when they look at a patient, but you never know. personalized medicine is really refining down to a molecular level an approach for a patient. And I think in a complicated disease like ME, that's the direction we're going. But we're going that direction actually in a lot of different areas. And oncology is probably the one that's leading the way the most. People have made the argument that there are much better outcomes. I think we're still proving that, but I think that's the direction that we're going. People have made the con argument that this actually costs more. Well, I think in the long term, particularly when you look at productivity of patients and their livelihoods, it actually costs significantly less. So we have, on the one hand, clinical medicine with sort of guidelines running headlong into genomics, larger scale databases that are being collected around patients in biotechnology, and these technologies and this concept is really they're crashing headlong together, and what we hope is coming out of this is sort of clinical medicine version 2.0, which is a much more individualized approach, which I think will serve the CFS-ME community very well. So what do we really need to do? Well, we need to look at diagnostics, and many of you this may be a familiar curve to many of you, certainly those of you who've spent any time in medicine. This is an ROC curve, a receiver operator curve, and what we use these for is to assess how well we're doing or how well a given test is doing at evaluating a function. So what you want to be is low false positive, so on the left of the scale for a test, and high true positive on the right of the scale. So you want to be as far up into that right-hand corner as you can possibly be. If you're that diagonal line down the middle, then you might as well flip a coin. You want to be high and arched. Most tests don't make it all the way into the corner. Actually, most medical tests are not that great. But the advent of some of this molecular level testing has really pushed a few very key areas farther up into that corner, and that's what we need to do with CFS-ME. What we really need to do is do that in many ways over time. So build many receiver-operator curves for many characteristics that we're collecting across patients, across every disease, across every condition within the disease. So David mentioned a few in terms of those that are tested by those assays, but we can add not only clinical criteria and patient information to that, but we can add genomic criteria to that. We can add molecular level testing to that, and we can do so over time and build sort of a massive array of information around that. So we're adding multiple levels and a lot of complexity to that, and then we can add interventions to that. So how do these tests perform over time? And this goes back to the outcomes issue that I was mentioning. So how are we going to do that? Well, going back to what I was talking about before, and excuse me because I tend to talk really fast, so hopefully you're all still with me here. There's a branch of medicine called Nosology, and it deals with how do we define diseases, how do we classify diseases. This is a branch of medicine that's obviously hundreds of years old and really hasn't changed all that much in those hundreds of years. And until the last maybe 50 years, we didn't really move it forward very far, and we're still using actually a lot of those classifications that were invented way long ago. And I think that's part of the reason why in CFS-ME we have such a hard time at defining what is our disease, because that classification was used for defining what really turned out to be unifactorial or maybe occasionally one or two factorial diseases, where CFS-ME very likely it has so many facets to it that it really defies some of this definition that we've built around that. So our goal is to build a stronger definition through clinical markers, lab markers, and biotechnology markers. I'm going to give you some examples of how we've done that already in other areas, particularly other infectious disease areas. But really the goal here is to show you that we can actually redefine some of these diseases, and some of the diseases you wouldn't really think about why would we really need to define them, but it turns out that it's actually extremely useful. And this is a methodology that we're now applying to CFS-ME with some success, and I hope we'll be able to show you some of that data very soon. So our premise in the Institute is that we can redefine all disease from a molecular perspective. It's just a question of finding the right molecules in a sense, and that's not always so easy. So the guy in the bottom corner is saying, offhand, I'd say you're suffering from an arrow through your head, but just to play it safe, I'm ordering a bunch of tests, and the patient is sitting sitting on the exam table with an arrow through his head. Well, that's kind of what we do in medicine. That's our approach. So we as physicians, we develop a differential diagnosis, and there's an infection in the patient. Okay, well, what do we start running through our head? Or the patient has a fever. What starts running through our head? Okay, infection. Well, you run down the list, and probably CFSME should be on there. Sarcoidosis, all kinds of things can go through our heads, and we start to try to whittle those down. Well, and then we start running a whole bunch of tests, right? We do a history and physical, which is for those of you who can't see the writing, it says, "I'm stumped, we'll have to wait for the autopsy." So the history and physical is really one way of running a bunch of tests. It's our physician's acumen in terms of extracting out some data points, but then we add a bunch of laboratory tests. Maybe we're doing some more sophisticated things in terms of bone marrow biopsies or bronchoscopies in more complicated cases. But really, we start adding to that armamentarium of things that we're starting to build out that array of ROC curves and data. And there's a better way. So this is a typical ICU patient, and there's a multitude of places where this patient could have had an infectious source entered, and this is probably should be twice as many arrows on here. But just to give you an idea of where a potential physician might look to start looking to define an infection or find a source for that fever. Well, we took a little bit of a different approach. We said, well, to heck with all that other stuff, because we're actually pretty lousy at looking at all of these places and telling you exactly why a patient has a fever, right? So patients in the ICU, they're in there for a few days to a week or more, and they develop a fever. Well, what do we do? Well, we start scratching our heads. Well, is that line infected? Oh, better start them on some antibiotics. The fever hasn't gone away yet. Let's get a bronchoscopy. Let's do, the tests start flying and the drugs start flying, and pretty soon we're down a road that maybe we didn't always want to go down. But there is a better way, and part of that way we set out to answer a very key translational clinical question. We said, here's a practical problem that physicians have in their daily workup and treatment of these patients. And how can we make that better? Well, it turns out that if you look at patients with fever in the ICU, you can actually cluster them. And on the right, I'm sorry about the projection on the color there. It looks like it's hard to read. But on the right hand side, you see differential time points in the columns of, I can't walk around. Sorry, I always walk around when I talk. Oh, is it a pointer? Oh, no, I just tend to wander when I talk. So the columns here represent differential times between how long the patient was in the ICU and what was going on with those. There's also a 0 time point that's not shown that these are all being compared to. But we basically took samples out of these patients, and what you can see is this first box here is patients with a line infection who we wound up, after the fact, definitively identifying a IV catheter line infection, a bacterial line infection in them. And you can see they have one particular set of signals turned on early on. The second big box is post-surgical patients. Well, they pretty classically will have this huge inflammatory response at 48 hours, and that's where their fever evolves. If you look further down, you see other boxes, which are corresponding to central nervous system bleeds, sinus infections, pneumonias. This really noisy one down here is a fungal infection. The pneumonia is this one here, which you can see is pretty active all the way across. So we can actually define profiles for these different conditions and define a molecular diagnostic that could be done at the time of the fever within an hour, and turn around and use that to say, okay, this patient doesn't have a line infection, no antibiotics for you, no line removal surgically for you, no, all kinds of changes in their clinical course. So I put this out there not to teach you about ICU medicine, but to point out that there is actually another way, and I'll run through some quick examples here. But the point is that we have to go back to sort of KOCHS postulates for defining whether this is an infectious disease, not sort of random criteria. We want to really go back and look at how do we define a disease, but we have to revise KOCHS postulates. So Koch was a famous microbiologist way back when, in the 1800s or so. But that's what we've laid the foundation of infectious diseases on ever since. And basically what he said was there has to be an organism there, and you have to be able to get that organism, and you have to be able to give that organism back into a body to generate that disease again. And we've sort of stepped away from that a little bit in medicine. But we have to redefine that a little bit for the 20th century and 21st century. And so we've come up with molecular KOCHS postulates. And the premise is that there is a unique dynamic host response signature for any given disease. The question just is, did you look in the right place? And I hope what I just went over helped convince you that that's true. Individual signatures may be predictive of susceptibility to disease, but they may also indicate a response to therapy as well as recovery and are time and state dependent. In other words, we can tell you not only the signatures that I showed you in terms of defining when a disease happens, but did we put you on the right antibiotic or not? Did we give you the right medicine to shift you back towards normal? And hopefully churning through all your brains right now is the idea that we can do this the very same thing in CFSME, which is exactly what we're up to. So we don't actually have to have specific pathogen signatures. We can get to that level in certain cases, but they're actually not required. But it turns out that we can, and many of you probably have seen some of the papers coming out on the proteomic side, we can start to differentiate some of these different clusters. There was a beautiful paper on spinal fluid between Lyme and CFS patients, and that was a great step in the right direction. I think you'll see a lot more to follow. So I'm going to diverge from that for a minute, and then I'll come back to some of the clinical points just to talk a little bit about what we've done in the network. And so we've, as we started this, we started the Institute with an eye towards collaboration, and because we knew we couldn't do it alone, and we didn't have a whole lot of money to do it alone anyway. So we said, we need some help. So we really looked to the community and the community physicians to participate in this network, and we've had some wonderful participation now from across the country, and indeed now even across the globe, and patients, of course, as well. So what we did as part of our infrastructure is set up an informatics-based infrastructure to collect data. So we're collecting data directly out of patient electronic medical records from physician offices. Sometimes we're providing the electronic medical record for the physician office. We help the clinician to gather data, but also to perform their daily job, so ordering the tests and ordering the medications and tracking all that stuff and tracking different criteria. That's a huge part of what we do, and in part of my previous life, as a point of disclosure, I started actually an EMR company to do exactly those kinds of things out of my PhD dissertation. So one of the key pieces is to be able to gather that data, but we can gather data from any source. So we have hospitals that we have direct data feeds from. When I was starting this, the previous work that I was pointing out was actually done in the Stanford ICU, and I had a direct feed off the vital signs monitors in the ICU that I would know when the patient had a fever before the nurse knew. And they would always wonder how we showed up with our little tubes to collect just as they were finding out the patient had a fever. And little pieces like that are a key piece to making some of these research projects work, and I'll talk a little bit about those at the end in terms of how we're doing that with CFSME. But the point is that we have both data and samples flowing between clinical sites and a central repository, and we're standardizing the way we collect those samples as well as the data. We need lots more. I mean, Dr. Bell did a lovely job at talking about some of the tools that we can, survey instruments that we can put towards patients to collect data, but we need to do a lot more in that direction in terms of not only standardizing, but validating some of those tests and standardizing the way we collect those across multiple sites. And then we've got lots of collaborations with different labs to process some of those samples. And, I think in any study, that's such a key point, is that you really have to look at the test type. So cytokines, for example, are a really difficult thing to measure, and it has a lot to do with the way they're collected. And if you don't collect cytokines in the right way, then your study is garbage. And you can put the data out there, but if it doesn't necessarily mean anything. So you have to be really careful about how you collect data. And we have huge problems with NK cell function assays, getting the clinician's office to send the right NK cell function that to the right lab, or at least to a lab that does it well and in a timely way, right? If those samples sit around too long, your data is no good. So we want to have both a good data collection mechanism, but also a quality check on that data. So we started an integrated registry and data warehouse that's fully integrated into our biobank. So far, we've just been running the biobank internally, but we're about to open that up to sort of more general participation. One of the key pieces to our network is that physicians can collaborate at any level. So they can do something as simple as pointing the patient to the network to get on our website and give us information or send us a sample along with that information. They can actually participate in drawing that sample directly and sending that sample to us. They can provide the sample directly to us as well as the data on paper or electronically. They can collaborate with us on a protocol, which is a little bit more involved than some physicians have room for, but we will help them do that and help us recruit for different studies and so forth. Or they can go all in, and we have full electronic access with them and full data sharing and biosampling, and they're actively participating in clinical trials, which is the place we like to have the most people, and indeed that's sort of the direction we're heading. Patients can participate at multiple levels in terms of, again, giving just data, giving medical records, giving blood or other samples, and so forth. So what we want to do is define a core set of information to be collected from different sites for CFSME, and then look at those in a large, across the larger population context, and start clustering patients together and start looking at, again, looking back at those outcomes and defining some of those data points, and we're well into that process. And we want to be able to enable the collection of data that goes outside those standards so that we don't lose that information, but we want to be able to account for it in any number of ways, and our system does that. So what do we need in the CFSME field? Well, we need cooperative patients. I think that's an easy one. We need collaborative providers. That's pretty easy too. We need scientific rigor. That's been, I think, slower in coming, but I think we're there in the last few years. We've seen some really high-quality researchers enter the field and some real good work coming out of it, and I think everyone's would agree that the rate of publications are increasing. We need some innovative thought, and I think that's slowly happening. We now have, obviously, core resources, our own and WPI and others. And, I put political will on here as optional. It's somewhat tongue-in-cheek. I think we would love to have political will. I think in the CFSME world, we've hit some walls, though, I think here as well as abroad, and, that yet needs to be addressed. But I think, actually, that we can address some of the problems in CFSME even without that. And they will come around, as they did in the AIDS epidemic, as they did in others, as we build evidence and build momentum. So a little bit more about our institute. We're as I said, in the middle of Silicon Valley. We're actually located on the campus of one of the leading hospitals in Silicon Valley, and probably the nation for that matter, and have a wonderful collaboration with them. We're helping run about 200 trials right now with partners, and in lots of different areas of medicine. And I think the only reason I mention that is not just to pat ourselves on the back, although I love doing that, but it is really to underscore that we need to be able to bring much more rigor and I hate to use the word legitimacy to CFSME, but we want to be able to demonstrate that the same techniques that we're applying in other areas of medicine apply equally well here, and really need to be used to the nth degree to really underscore that this is a real problem, because it is a real problem. And so we're going at that in a number of ways with CFSME and neuroimmune disease in general as a key cornerstone to our research efforts. We currently have 4 trials that are in pilot phase, and several that are several more behind that, and are very excited about some of the opportunities. So what's in it for a clinician in terms of joining our network? Well, hopefully better knowledge through all of this, potentially better practice patterns within their practice if they would like to do that. lots of collaboration and more referrals, participation in clinical trials, and strength in numbers. I think that's one of the things that we've seen is key, whether you're talking about addressing something in CFSME or in the healthcare system in general. If you're a single physician standing alone, you're not going to get very far these days. So a lot of our time is spent gathering ideas from physicians and pooling people together and going back. You know, we have a problem with hospital X. Well, if Dr. Smith says something, well, maybe it's going to happen, but if we get 50 doctors together who all go to the same hospital, we're going to get a little farther, and I think the same goes for our political will question. What's in it for the patient? Well, you get to help us drive research forward, which is a key desire, I think, of all of us. Hopefully some answers to some of your personal symptoms and problems. Ultimately, I think better care for everybody and potentially connection to other patients and providers that you may not have had otherwise. What's in it for the researcher? Well, definitely better data and a lot more of it. And that's, I think, one of the key points here, and a lot more outcomes focus, so a lot more, I think, scientific rigor in terms of the quality of data and so forth that can be put underneath this, as well as focusing on the standardization. And access to a lot of resources that would otherwise be unavailable. We have wonderful collaborations with Stanford and UC and others. The Stanford Genome Technology Center and Dr. Ron Davis are right down the street from us and have been wonderful collaborators bringing to us stuff that isn't even available on the market yet in terms of technologies to run. So we're doing actually gene sequencing now. So that was one of the genome sequencing centers, and they're now running CFSME patients, I'll be happy to report. And we're also using some differential spliced arrays and so forth, so we'll have a lot of interesting data coming out in the very near future. Collaboration is a biggie, and I think that's one of our cornerstones, and what I like to call benevolently guided research, when I can say it. So the network really is designed as a machine with multiple ports of entry. As I mentioned, we have electronic medical records directly from the provider that can be, it's got a little mangled here, sorry, that goes straight into our data warehouse or our health information exchange. Data that's entered by patients can come straight in, so we have an iPhone application, or any smartphone really can get on it. We have an iPad application too now, so patients can enter data directly. We also are real big on sticking devices on patients. I didn't bring one with me, but we have these little essentially fancy pedometers that sync to directly into our database. So every time a patient walks by their computer, it syncs their activity level for the last day or so up to the computer, actually the last week for that matter. So we can get minute-to-minute activity levels for patients without them even having to think about it, and it puts some more quantitative measures on some of the metrics that Dr. Bell was talking about. And then, of course, we want to have labs and pharmacies and so forth fully integrated into here. So this gives us an ability to track patients, to track them in terms of what medications or herbals or other over-the-counter medicines they're on, to track them in terms of what other interventions are going on, how they're doing, even if we don't see them in the office. And I think that's one of the big failings of medicine across the board is, we see a patient, okay, we see them, maybe they come back in a week, maybe they come back in a month, maybe it's 6 months before we see them again. And then we have, half hour with them maybe, and we're supposed to get a snapshot of the last 6 months. Well, you basically wind up hearing what the patient wants to tell you, and then maybe a little bit about what you specifically wanted to dig into. This can give us so much more in terms of the patient portal of the kinds of information and a summary of that information than a patient often can summarize themselves. It's great to have the patient input on these too, because that gives the patient a direct communication channel into the physician, and when they trigger certain things, those are instant sort of red flags. On the back end of all of this network, we have a de-identified research database, and these are opt-in things, by the way, for patients, so if you don't want to participate in certain kinds of things, you don't have to. And then we have an active clinical trials research database for active clinical trials, so researchers can go in and look at what's going on currently, or they can look at sort of the summary view in a de-identified sense. But we like to keep this as black box to everybody as possible, because most people don't want to know what the innards look like. So for most people, it's just, I picked up my phone, or I just stuck on my belt clip this morning, and I'm on my way, or I used the web interface. So most of what we do is via the web, which works well for almost everybody. There's a provider portal, there's a patient portal, and as I mentioned, some of the social networking aspects. On the back end, we have a lot of different statistical and bioinformatics tools built into the system, and we're constantly sort of revising and mining the data that's there, as well as tracking all the specimens that we have flying around. I'm not going to spend too much time on it because I'm running out of time here, so I'm just going to plow on through just to give you an idea. We can take in scanned information. I mentioned already sort of iPads and other data feeds into the system. Our system tracks sample kits that can go out to our providers. We send them out in bulk to the providers, but we can also send them individually to patients and get them back from their physician or from the lab directly. We recently had our first whole body donation, which had some issues with it, but the pathologists weren't all that cooperative. But on the whole, it was an interesting process, and one we learned from. But I expect that there'll be more. But we can pretty much take in any kind of tissue, which is really exciting. So where are we going with all this? Well, we want to do some large-scale studies in terms of clinical data gathering, as well as clinical trials, and we're actively on our way to that. We want to create and continue to expand our biospecimen collection, which is, as far as I know, is the largest, at least clinically documented biospecimen repository on CFSME, and continue to expand the amount of RNA, DNA, and other molecular testing that we're doing, and continue to gather normals as well, because I think that's a key piece that we're missing here. I'll stop for questions. Thank you very much. Invite you to join us.
This is working out quite nicely. We want to get away for coffee because you all need it, but we've a little bit of time, so we thought we could move some of the questions forward, particularly for the last three speakers, because the first speakers get lost towards the end of the day if we're not careful. So if there are any questions, I've not received any at the moment for all these first speakers. It does seem to me, Ian, yes, go on.
Showed the genomic profiles. I guess that was one patient within a few hours, days, and so on. And there was quite some dramatic things. Are you saying that there can be spurious infections of that patient, which will give you different genomic profiles? What are you actually saying from that data?
So that data was actually multiple patients. It was sort of a summary view of multiple patients and the patterns that go along with particular conditions. What the point really was that we can identify, kind of Jonathan Kerr's approach in terms of his paper a while back, that we can identify a limited set of genes that have changed in a given state in the body, not just a different, not just a specific condition. So pneumonia on day 1 looks different from pneumonia on day 2, looks different from pneumonia on antibiotics, looks different from patient with resolved pneumonia. And so the point for CFS is, of course, that there are multiple stages as well, but more importantly to the point is that at that baseline, we want to be able to differentiate a CFS signature, if there is such a thing, and Jonathan's data would indicate that there are probably several clusters of signatures, so potentially several different entities, disease entities that are similar in terms of clinical picture, but actually potentially different on a molecular level. And that actually points us towards different treatment paradigms as well.
But I mean, how realistic is that? If there's variation, say, you've got 50 patients, you have 50 different patterns at 4 hours, 8 hours, 4 days, what the hell does that mean, actually? You'll never find a biomarker that's any good for any individual if it's varying all over the place.
You're absolutely right.
Like.
You will get nowhere with 50 patients. You have to do a lot of patients, and you have to start teasing these signals out over a large cohort to be able to actually separate the signal from the noise. So you're absolutely right. And some of the signals you may not be able to pull out of blood. So we when I was still at Stanford, we did a beautiful study looking at gastric biopsies in H. pylori infections, and you could sample normal mucosa 2 centimeters away. You could sample an H. pylori ulcer, and the signatures could not be more different. And further, if that H. pylori ulcer progresses to a precancerous state, the signature is different yet again. If you treat it with antibiotics, the signature starts to resolve and improves and converts back to normal or a near normal state. And if it goes to cancerous, obviously that's yet another state.
But you still believe molecular markers are the answer with all that variation? Why don't you concentrate on the environment more? Instead of all this fancy dance stuff with PCR and complicated, expensive machines, which sounds very good and very scientific. We all know that. But it doesn't do much good for the patient.
It does if you can extract out a marker, and that's really the question. So you're asking absolutely the right question. If we can extract out a molecular signature, even for a subset of CFS patients, that takes that subset out of the pie that is CFS and says these patients are definable by this criteria. Forget your Canadian Fukuda, whatever. These molecular criteria, I can develop a PCR test to identify those patients. And then potentially the next step is, well, if they have these X, Y, and Z genes abnormal, well, what are we treating there? And what are other pathogens? Yeah. Well, it's a hope that we do have some initial data on, and I think it's going in the right direction. And Jonathan Kerr's data, I think, would underscore that.
Thank you. Thank you. Yes, sir.
I have a question for David Bell. You talked about the groups with remitting illness and persistent ME, and I guess you just got a snapshot. How interchangeable are these groups? How fixed are the groups?
I think that's an excellent question. There is a small group that I think fluctuates between remittent and the severe. But in general, the severe is a pretty fixed group, and they stay very severe. The only way that I'm aware of determining that really severe disabled group is by looking at the severity at the onset. So that in general, all of those people who stayed severe were bedridden or housebound for the first 2 years of their illness. In the remitting group, most of them were able to go to school and, they'd have some prolonged absences, but they were nowhere near as severe at the beginning. There are a couple of other papers that have hinted that the prognosis is based upon initial severity, and I think that that's something that is important. But in general, the groups are somewhat fixed, at least the disabled group, the very disabled group.
Hello, is it on? Yeah. I'm Trish Groves. I'm Deputy Editor of the British Medical Journal, BMJ. So thank you very much for inviting me today.
Thank you for coming.
There's a question for Dr. Kogelnik. I'm sorry, I didn't catch how to say your name. I'm really sorry.
Perfectly fine.
Okay.
Kogelnik.
Kogelnik, okay. You mentioned that you are doing clinical trials, but I wasn't entirely sure whether you've got any randomized controlled trials on these conditions, on chronic fatigue syndrome and ME yet. It sounds like you're really quite a long way from that at the moment. Is that right?
Actually, we have 2 randomized trials. There's some pilot trials that are currently underway. There's 2 randomized trials we hope we'll have first patient in within the next 6 months.
Oh, great. Okay, well,
and a third right behind it.
Just to clarify, because the term clinical trial tends to mean many things, and
it does.
What we really mean if we're trying to find a way to burst this bubble and find treatments that work is you actually need trials where...
Big fan of randomization and placebo control.
You have people split into groups randomly, and you've got an intervention. You've got something to offer, and for that you need a clear question that you're trying to answer. And as you quite rightly say, you need very defined groups of people. So I'm interested that you've started those. That's great. Are they registered? And if so, I'd love to know more details.
Well, as I said, first patient in hasn't happened yet for those trials. So as we approach that point, we will register them.
Right, you have to register them before you recruit. You know that, don't you?
Well,
yeah.
We need to finish the protocols
first.
Yeah.
So you're near to that. But that's terrific because, as many of you know, that when clinical trials are started, these days you have to register them in a public registry, which is great because then people can see what study you're planning to do. They can come along and join the study either as researchers or participants in the trial, patients. So that's terrific. Looking forward to seeing those. Thanks.
Technically, I think you only have to register them if they're government funded, but we'd be happy to register them anyway.
Yeah, no, sure. You're right. In America, you have to register with clinicaltrials.gov if you're going to produce, you want a product that's going to get onto the market in the States. I appreciate that. But you'll also find that you won't be able to publish the work in 800 of the major journals in the world unless the study was publicly registered before the first patient's recruited, because journals are absolutely insisting on trial registration. For all the reasons I've said, it's very important that patients and researchers are aware of the trials that are underway. Thanks.
I agree.
This is Jean Monroe. A question for Dr. Kogelnik, if I may, please. It's to do with the last question, in fact. It's extremely difficult to get to the point of having a clinical trial registered unless one's got research funding and a rather broad spectrum of support. The difficulty is in this country, there are very few people working with patients with chronic fatigue, apart from those who are given the license to do so, which means that they're usually psychiatric institutions with pacing and other things of that sort going on. Would you think that you could audit work from individuals and from clinics where they may have quite a lot of data? Because publishing of audit might be possible as an alternative to a clinical trial.
Yeah, so part of what we've set out to do with the biobank and the registry and the database as a whole is really be able to do what the woman from BMJ was speaking about are prospective trials, and what one of the things we would like to be able to do is what you're suggesting, I think, is more of a retrospective trial in terms of data analysis and data mining, and absolutely we are doing that already, and it's, I think, yielding good directions for us and for the field as a whole. But for sure, that's something that we want to do and we want to enable, and that's part of the reason for the standardization as well. We need to be able to have all that data coming into some kind of repository so that when patient X says they have a cough, it is somehow defined the same way as patient Y saying that they have a cough, and so on down the list of criteria. So did that answer your question?
Thank you.
It's a question, comment for Andreas. For many diseases, animal models have been developed. You have to use them with great care, but nevertheless, that brings in a lot of good scientists who can do the kinds of experiments that you can't do on humans. Unfortunately, this disease seems to be so complex that I imagine it's very difficult to even begin to think of an animal model. But is anybody trying to develop one, and could this be a possibility, and would this then make it easier to recruit lots of good scientists into the field?
I think if we had an animal model, that would be wonderful, as you suggest. I'm sure there are some folks, and I've heard from different folks about sort of the same issue in trying to find one, but I think part of the problem in finding an animal model is, as you indicate, the sort of the multi-system, multi-faceted aspect to the disease. I think we can reach some success in terms of carving off pieces, maybe a gut animal model versus there's a few others that we could think of, but as a disease as a whole, I think it's going to be really hard to create an animal model for the disease.
Thank you. Thank you.
Hi, good morning, Dr. Mandy Platts. I've worked in general practice for 30 years, and I've seen exactly what Dr. Bell was talking about. And I think it links to the clinical definition that then the second speaker spoke about, with this 20% of really serious from the very beginning, serious response, can't do anything at all, who don't do well, and the 80% who actually do incredibly well in the sense of your health perception. They think they've got better, but they haven't totally. And I just wondered whether that is muddling up and making difficult this research and the definition and the political perception, because these two groups are so incredibly different, with different outcomes, different lives in the end, and different sort of beliefs about their illnesses.
Thank you for that comment. At one point, I was working as chairman of the advisory group for the United States government. And at that meeting, we had Social Security and NIH and CDC. Social Security person who was in charge of the disability at one meeting stood up and said, "We're very proud. We now have over 25 people who have received disability for ME, or for chronic fatigue syndrome." 25. And he was proud of that. Now, I'm not exactly sure that was the number, but I was aghast because it's still not recognized except in a very, very small minority, even a minority of that 20%. Most of the people who do go on, who are in that intermediate range and cannot do sustaining work, they have to be diagnosed with something else that's considered legitimate disabling illness. And I think that that is a very big problem. The fact that the head of the disability was proud of the tiny number that they have approved for disability is really terrible.
I think to add to that, it's really the criteria that's the problem. And so we would like to see that criteria evaluated and changed to more appropriate criteria. You think about the people who are completely bedridden and they're supposed to get up on a treadmill and prove that they have a VO2 max below normal, which is impossible. So I've looked at that, and I know that there is considerable work that needs to be done. So
Thank you.